By September 2024, there were over 20,000 cases of Mpox reported and 600 deaths according to data from World Health Organization. The testing rates for Mpox are still relatively low at 40%, meaning there could be underreporting from countries that are currently impacted. The Mpox virus has been recorded in more than 120 countries and territories since 2022. The WHO declared Mpox in the Democratic Republic of Congo and neighboring countries a public health emergency of international concern, the WHO's highest level alert. This declaration was made due to the ongoing and rapid spread of the new virus strain in DRC named Clade 1b. The majority of cases and deaths are among people under 15 years old, and 73% of them are male. The epicenter of the ongoing outbreak and most deaths are in the Democratic Republic of Congo. So, what's the current vaccine development and challenges situation? Mpox is a viral infection for which there's no specific treatment that is available. While the Mpox virus continues to pose a significant public health threat, particularly in Africa, progress in both vaccine development and treatment options offer a promising path forward for outbreak management. Several vaccines are currently under development and some have been approved for Mpox prevention. However, the capacity to produce these vaccines at large scale, especially in the countries with the highest burden of Mpox, remain a significant challenge, as highlighted during the 2022 outbreak that occurred globally. Production delays during previous outbreaks underscored the importance of diversifying vaccine platforms, including mRNA vaccines, which offer scalability advantages. Combining novel vaccine platforms such as the mRNA and more traditional options provide a broader toolkit for combating this virus. Continued investment in clinical trials, production infrastructure, and equitable access and distribution will be essential to controlling the ongoing outbreak and preventing future resurgences. So, let's look at the vaccines that have been approved or under development for Mpox. The first vaccine is Jynneos. Jynneos is a modified Yersinia Ankara, abbreviated MVA. It's a live, non-replicating vaccine developed by Bavarian Nordic. It is FDA and European Medicine Agency approved for the prevention of both smallpox as well as Mpox. Jynneos is a licensed product in the US, Canada, UK, European Union, and Switzerland. As of the time of recording this presentation, the vaccine is approved for use in adults 18 years of age and older for the prevention of smallpox, Mpox, and other orthopox diseases. Standard administration of Jynneos is a regimen of 2 doses administered 4 weeks apart. The World Health Organization recently pre-qualified this vaccine in September this year actually, which paves the way for wider use in low and middle income countries, particularly in the ongoing Africa's widening Mpox outbreak. The second vaccine is ECAM 2000 smallpox vaccine. ECAM 2000 smallpox vaccine is a live Yersinia virus, a vaccine developed by Emergent Biosolutions and indicated for active immunization against smallpox disease for persons determined to be at high risk for smallpox infection. The vaccine is FDA authorized for emergency use against Mpox as well. Although it provides a robust immunity, ECAM 2000 vaccine contains live replicating Yersinia virus, and its administration to individuals who have immunosuppression or immunodeficient syndromes may result in Yersinia complications, including fatal outcomes. Individuals with severe immunodeficiency who are not expected to benefit from this vaccine should not receive it. These individuals may include people who are undergoing bone marrow transplantation, or individuals with primary or acquired immune deficiency syndromes such as HIV/AIDS. The third vaccine is Moderna's mRNA 1769. The mRNA 1769 is an mRNA lipid nanoparticle vaccine expressing Mpox surface proteins. It's a multivalent vaccine with 4 well-defined orthopox virus protein targets defined following deep immune profiling of the protective Yersinia virus-induced immune responses. This vaccine candidate is currently in phase 1/2 trial with about 351 healthy adult participants who are enrolled in the UK. The data from the phase 1/2 trial will be out around mid-2025 and will help to shape the phase 3 trial design. If determined safe and efficacious, Moderna's mRNA 1769 will add to a diversified vaccine platform, including the mRNA vaccines, which offer scalability advantages and address production delays. The fourth vaccine is BioNTech's BNT166. BioNTech is developing mRNA-based Mpox vaccine currently in early clinical trials with demonstrated protection in preclinical studies involving mice. 2 multivalent mRNA vaccines were tested in preclinical studies to target different viral forms and expand the range of immune response. One vaccine, BNT166A, contained up to 4 viral antigens, namely A35, B6, M1, H3, while the other, BNT166C, contained 3 antigens, A35, B6, and M1. Both vaccines generated strong T-cell responses and IgG antibodies in mice, including antibodies capable of neutralizing both Mpox viruses and the Yersinia virus as well. In challenge experiments, both BNT166A and BNT166C provided complete protection against Yersinia clade 1 and clade 2b Mpox viruses. The fifth vaccine is TNX-801. So the TNX-801 is developed by Tonix Pharmaceuticals. It's a synthetic live virus vaccine that is currently in preclinical development for Mpox and smallpox prevention. Phase 1 trials are planned to start later this year. The sixth vaccine is KM Biologics LC16. The Japan's LC16 vaccine is a modified Yersinia Ankara vaccine, just like the other previously described platforms. It's developed by KM Biologics and has secured domestic Japanese approval during the previous Mpox outbreak in 2022. The World Health Organization recommends its use, but it has not yet secured a global regulatory approval. Limited use in clinical trial for this particular vaccine in the prevention of Mpox in children aged about 2 to 12 years in the DRC and Uganda is documented. The seventh vaccine is C16M8. This is a live attenuated smallpox vaccine derived from the Lister strain of Yersinia virus developed by the Chiba Serum Institute in Japan in late 70s and approved in the 1980 according to the procedures of the Japanese regulatory authority. It's stockpiled in Japan as part of its national preparedness and under study in the US for Mpox. LC16M8 is administered as a single dose using the traditional scarification method. The eighth vaccine is Sinopharm Mpox vaccine. The vaccine is independently developed by the Shanghai Institute of Biological Products, a subsidiary of the Chinese National Pharmaceutical Group Sinopharm, and the vaccine is based on a live attenuated orthopox virus modified Yersinia Ankara. According to the institute, the MVA strain has proved its safety and efficacy as a potential candidate vector for vaccination. The MVA Mpox vaccine from Sinopharm is produced using mature cell factory production process, which according to the developers is stable and reliable in quality. Preclinical studies have shown that the vaccine is safe and immunogenic against the Mpox virus in non-human primate models. There are other multiple vaccine candidates for Mpox that are currently in preclinical stages of development. These include Onconetic, which is a subunit vaccine, Nuvax, which is a viral vaccine, Imagex vaccine, which is a synthetic peptide Mpox vaccine. And in terms of the future outlook for Mpox vaccination, the global health community is focused on improving vaccine availability and expanding immunization coverage, particularly in regions most affected by Mpox. Ring vaccination strategies targeting high-risk contacts have proved to be effective in previous outbreaks and is likely to be replicated for the ongoing outbreaks. However, equitable vaccine access remains a significant challenge, especially in resource-limited countries. Thank you for watching and see you in the next one.